All right, so thank you. I know a number of you have already heard this. It's really strange. I've got some of my incredible collaborators, Bridget Seitz, Edmund, Tom, and Greg in the room. So I'm going to invite anybody who's been a part of this to speak up anytime that you want. I would really rather this be a discussion than me telling you what's happened. So many of you have seen this before, but I hope to build a story for you as to why 5-HT7 receptor agonists are compounds that should be developed and could be clinically relevant. Before doing that, though, I want to give you the background story as to why this is so. And this is really my most favorite side. These are the people who helped make this happen. This is my team. As a couple of years ago, Bridget Seitz, who will become hopefully Dr. Bridget Seitz next year, has done a majority of the in vivo work here, and she's been a partner in this with me for a long time. Greg Fink has as well. Janice Thompson and Emma Darius have been important in the molecular work you're going to see. In terms of the drug discovery work you're going to see, Edmund Ellsworth, Tom Dexheimer, and Rick Newbig has helped me be brave in places that I have not been in the past in moving this idea forward. So stop me if you have any questions, okay? I'd really rather us be together on this. So this is the structure of 5-HT. It was discovered in 1949 by this gentleman, Maurice Rapport, who I actually had the ability to meet. He and Green. 5-HT is made primarily in the enterochromaffin cells of your gut. About 90% of your body load is from here on down, and about 10% is from here on up. Your circulating 5-HT is typically taken up in the platelets. Right? So you've got a lot of 5-HT working through your circulatory system, but most of which is contained and is not biologically active. 5-CT is a compound that we're going to use as a 5-HT agonist. Right? So serotonin has been around a long time. And it was discovered because of work that could be done in things like this. Right? For those of you who know Carrie Northcott drew this when she did her PhD way back when, about 18 years ago, and I love using this. This is the isolated tissue bath. Right? You can put smooth muscle of really any kind from any species and measure the isometric contraction or relaxation. So kept warm by a jacket and aerated by this frit at the bottom. What you can do is place this on a stainless steel rod here, add a transducer on the other end, and you can add things to this bath. Okay? This is one of the ways that pharmacology has moved forward, is through isolated tissue bath assays like this. If you ever go to London, you can go to the British Museum and go and worship Sir Henry Dale's first isolated tissue bath. I have done that. I genuflected appropriately because this really helped change my life. The power of this is phenomenal. Right? It looks simple, but what it can do is amazing. Right? This, then, is how we know 5-HT. If you take increasing concentrations of 5-HT, add it to that tissue bath, and you measure force, virtually every blood vessel from the mouse all the way to the human that you put in is going to do what I show you here. It's going to contract. Okay? If you add 5-HT, you get a concentration-dependent contraction. So the history through decades of research has been that 5-HT is a vasoconstrictor. Now, let's take into account this piece of information. Now let's take 5-HT and look at its ability to contract blood vessels, not only in normal animals, but in animals that have high blood pressure. In this case, we have a isolated tissue bath experiment where we're using the rat superior mesenteric artery. So here again is 5-HT, and we're measuring contraction as a percentage of phenylephrine. What we've done here is to take an artery from the sham. This would be an animal with normal blood pressure. And then an animal that is deoxycorticosterone acetate or docosalt hypertensive. This is a mineralocorticoid-based model of hypertension. What you see is, even my English professor husband can see the difference between these curves, right? They're different. 5-HT has a lower threshold. 5-HT has a higher potency, and it has a higher efficacy in causing contraction. So put those two things together. 5-HT is a vasoconstrictor, and it's a better vasoconstrictor in hypertension. It was those ideas with more than 15 years ago led us to this hypothesis. Right? And this was really done in conjunction with Greg in being brave and moving in vivo in ways that my lab hadn't yet at this point. Right? And that's 5-HT. When you give it in vivo, it should elevate blood pressure and do so more efficaciously in hypertensive rats. That is a logical hypothesis to put forward, given everything that I've said. Right? So that's the idea. And here's the beauty of what in vivo protocols can do. This is that very protocol where we have the sham animals I described and animals which, like the sham, are uninophrectomized, but also receive this mineralocorticoid dopa, and they're put on salt. Right? We wait 21 days for this to develop, right? And they do. They get pretty hypertensive during this time. And at this point, we can put in what's called a radiotelemeter. This is going to allow us in a conscious way to measure the blood pressure, heart rate, the activity, and even the temperature of the animals. 1 week after that, then we put in another instrument, and this is an Alzheimer's mini pump. And this pump, when it's filled, will release through osmotic pressure either vehicle or 5-HT. Where this protocol differs very much from what's been done in the many things published before it is we don't give 5-HT for just 4 minutes or 10 minutes. We give it for a week. Right? We want to understand the long-term effects that 5-HT could potentially have on blood pressure, because blood pressure, while it is measured and regulated every second and minute, hypertension is a chronic problem. Blood pressure regulation is something that needs to be chronically considered, and thus the week that we have of this protocol is what we wanted to do. Once that's done, then we can take the blood, measure free 5-HT, measure platelet 5-HT, and measure a whole lot of things as well. What I'll tell you is that this model of 5-HT, this infusion at a dose of 25 micrograms per kilogram per minute, increases blood 5-HT from about 3 to 5 nanomolar to about 20 to 30 nanomolar. So what we've done is increase the amount of free 5-HT that's available. It increases what's in the platelet a whole lot, because the body all of a sudden has all this extra 5-HT that it needs to do something with. That's increased as well, but what's free is increased. Right? So what was my hypothesis? Right? That 5-HT would cause a hypertension. All right, you know what's coming. Right? I was completely, utterly wrong. These two people I owe incredible thanks to. Jessica Diaz was a graduate student in my lab at the time. Andrew King was a graduate student in Greg Fink's laboratory, and they worked together to do this experiment. They set this up as such. They measured over time in days, right? This is not hours, this is days, right? Where C is control, S is when serotonin was administered, and on the y-axis is arterial blood pressure. These measures in these animals are in the conscious state. So you see two groups of animals, and that's exactly how it should be. At the bottom, these are the animals that are sham. They have normal blood pressure. They took just about 100 millimeters of mercury, just like we do. And in the black up above here, these are our dopa salt. So this is a profound hypertension these animals have. At this hatched line, let's start that infusion. That's where the osmotic mini pump is given. And what you see is if you infuse saline in the black, it's really very stable, right? That's a wonderful thing, both in the dopa and the sham. If, however, you put a pump that's got this 5-HT in it, now you see not an elevation in blood pressure, but a fall. Not only in the normal, but you nearly normalize the blood pressure of the hypertensive animal. And it stays down for 7 days out. So now I've completely disproven my hypothesis, right, and need to rethink about what serotonin can really do. Right? This was fantastic, right? Disturbing, confusing. Jessica came up and said, Steph, I'm pretty sure I did this wrong because blood pressure didn't go up, it went down. I said, let's do it again and see what other people find. And repeatedly, this particular observation has been seen in about half a dozen different investigators' hands at least 2, maybe 3 different institutions. So it's something that I feel solid and confident in being able to say that this dose of 5-HT causes this fall in blood pressure. That's remarkable. What we were able to do then is have Patrick Davis, who is now in his almost last year of residency as a surgeon at Duke, is do this. He took a different kind of pump. Yes?
Steph, this response you get, what concentration did you say did 5-HT?
25 micrograms per
No, what was
kilogram
the
per minute.
concentration in the blood?
Yeah, so from about 5 nanomolar to about 25 to 30
And what does
nanomolar.
25 nanomolar do to an isolated vessel?
What, you're going to see that. Okay. Promise. Right?
Set you up beautifully. I'll take the 20 bucks afterwards.
All right, so here's a different kind of pump. That osmotic pump that Bridget has used, what, hundreds of, thousands of at this point, is only going to let you put in 1 dose. And once it starts going, it starts going. This pump, the iApprecio pump, which the Fink lab and my lab a little bit demoed and really helped do the beta testing on, lets you fill this little blue reservoir with fluid. And this machine is a pump which can be programmed such that you can deliver doses at particular times, and you can change the flow that comes from this pump such that you can change the dose. So you can do what is a dose response curve in an animal. Our hesitancy for using these pumps a lot right now is because these pumps, well, last we checked, the outlet pumps were about $30 a piece. These were over $300 a piece, right? And you can't reuse them. So it's still powerful though. And what Patrick did is to take iApprecio pumps, fill them with 5-HT, but now do what amounts to a dose response curve. And I want you to focus here just on the pink boxes. On the x-axis is time in days, not hours. On the y-axis is mean arterial pressure. And using animals that have been planted with the radiotelemetry, these conscious blood pressures show that as we increase the dose of 5-HT, we get a fall in blood pressure that's dose dependent. What we realized with this experiment is we probably didn't wait long enough between these steps. And so that was a very real learning experience for us. So now we've got two different investigators who have done this. So let's go back and think about blood pressure. If blood pressure falls, how does that occur? What we know about blood pressure is that total peripheral resistance, that's TPR, and cardiac output. The geometric mean of those two is what makes blood pressure. Both of those variables can be controlled by different parts of the circulation. So arterial smooth muscle tone is unquestionably important for total peripheral resistance. Just how constricted, how dilated, how open your small arteries and arterioles are will determine the resistance to blood flow. Differently, as Greg Spink's program Project Grant has talked about for the last 15 years, venous smooth muscle tone can contribute to cardiac output by assisting heart filling through venous return. Both of these sides of the circulation are innervated by the sympathetic nervous system. So what we have right here are two different things that we can test. The ability of the sympathetic nervous system to control the circulations and blood pressure, and then the separate ability to investigate the arterial and venous smooth muscle contribution to blood pressure. So that's what we're going to do. We had, though, to really figure out what we should pursue in a focused way, and here then is where collaborators are fantastic. This is Dr. Carrie Scroggin. She's at Loyola University. We sent her those Alzette pumps blinded. So they were just filled and she didn't know what they were filled with. And I asked her, would you put these in rats and tell me what you see? And what she could do that we couldn't at that time is measure cardiac output. By putting a flow probe around the descending aorta, not only was she able to measure blood pressure, but because she could measure cardiac output, we could calculate total peripheral resistance. Remember, blood pressure is the geometric mean of these two. What she found is, lo and behold, 5-HT, not exactly the same time frame, it lowered blood pressure. Okay? It elevated cardiac output, but boy, did it lower total peripheral resistance. Okay? We have seen this in our own hands in different ways. So that was a place to go to think, it's got to be arteries. If you're going to lower TPR, you have to dilate arteries. So where do you go in the body? What arteries do you take a look at? So those of you who know me have heard this before, but I have to tell it this way. This led to the smelliest experiment I've ever done in my life. Right? Bridget was with me all the way along. We cleared the lab when we did this experiment. We took animals that received that vehicle of 5-HT, gave it to them for 24 hours, and then gave microspheres that were fluorescent. And what those microspheres would do is lodge in places where there were greater flow. So where there were more microspheres and we could reclaim them, there's more flow. What you have to do is take out all the parts of the animals and you dissolve them in potassium hydroxide at 60 degrees Celsius. It is vile. There is no other way to describe it. Have to do it. So we decided we were going to bite the bullet and do it. We did for at least 12 different experiments, 7 to 10 that I'm going to show you here. I'll never forget the smell. But what we did is found that it was in splanchnic organs. And in this case, the spleen, when you compare flow between vehicle and those that received 5-HT for 24 hours, and the small intestine. So that helped us say, well, we have to start somewhere. And looking what 5-HT can do arteries, let's start here. Okay. So we hung up all different kinds of mesenteric arteries. Superior mesenteric arteries, smaller mesenteric arteries. We went back and hung up the aorta, went and hung up the femoral. I've hung up just about every artery I know how to do. I cannot observe 5-HT cause a relaxation of arterial smooth muscle. Cannot see it. Right? Have had Patrick do that for his thesis. He could not see it. Right? Noted, we have only gone down to blood vessels that are about as small as 250 microns in diameter. So one of the concerns I have is that we're missing vessels that are even smaller than that really could play a role in TPR. But that said, this surely is not happening. So if it's not arteries, is it the sympathetic nervous system? Are we causing a fall in TPR? Not because we're directly relaxing the arteries, because we're pulling away the sympathetic tone that is important to keeping TPR low. Let me make sure. Keeping the arteries contracted. So we did two different types of experiments. With Sue Barman, a member of this department, we infused 5-HT into anesthetized rats and actually measured Splanchnic nerve activity, preganglionic and postganglionic. 5-HT did nothing to it. With Emma Darius in the laboratory, she hung up blood vessels and turned on the sympathetic nervous system through electric field stimulation. Right? That causes contraction. There too, 5-HT did not change what the sympathetic nervous system does. So I am batting 0 at this point. Okay? 5-HT is not relaxing the arteries. 5-HT is not inhibiting the sympathetic nervous activity or the nerve activity to this bed that we think is important. So we got to go to the other place that we think is important to blood pressure, and that's to veins. So this is a picture of the rat venous circulation, most of it at least. What you see here are two sorts of circulations. You've got one that lies along the spine, and then you have a special hepatoportal circulation here. So this is the liver. Here is going to be the portal vein, and here is most of the mesenteric veins, including this superior mesenteric vein. We're going to take all of these veins, this vein here, the abdominal vena cava, the portal vein, and the SMV, to understand whether or not these particular vessels might relax. Now, there's no question veins don't control TPR in the ways that arteries do. They contribute maybe 10 to 12% of what's in the body. So they do control some, but not like arteries. So here's what we saw. So Peter, this is your question, right? We took isolated superior mesenteric vessels. And in this case, it's the paired artery in the red and the vein in the blue. Graphed either as from baseline tone, that's going to be the arterial experiment, or contracted endothelin, that's going to be the vein. We looked at what the effect of 5-HT was, and it was really interesting. Right? This is a simple experiment, but it was incredibly powerful in what we could conclude from it. 5-HT, even if you contract the artery ahead of time with endothelin, it causes contraction. We could not see it cause relaxation. Now when you contract the vein and you give it 5-HT, you see right here, along concentrations that we can achieve in the blood, right, about 30 nanomolar, the vein is relaxing. And it relaxes in a concentration-dependent manner until you get up to pretty high concentrations, and here is where both vessels take off. Right? As a receptor pharmacologist, what we've been able to demonstrate is this is likely due to activation of 5-HT2A receptors. Right? But it has to be that there's another receptor that's involved here in how 5-HT is causing venous relaxation. Okay. You buy it so far? Okay, so what is that? Right. Well, we'll get to that, but one of the things I wanted to show you is that I think there may be the potential in humans. Through the program project grant here that Greg Fink lives, we've been able to get samples of bariatric specimens. These would be small intestines, and I have taken out the artery and the vein here and mounted it in the same kind of isolated tissue bath that I showed you. This is just one experiment that we have, demonstrating here that now a different agonist that has affinity for particular serotonin receptors, 5-CT, it contracts the artery and it relaxes the vein. So what I hope to, if I could get more samples, is demonstrate that in fact, what we're seeing in the rat is something that we're seeing in the human. This is the receptor we think is responsible for this relaxation. The 5-HT7 receptor was the last cloned receptor. It is a heptahelical GS-coupled receptor. And because it's coupled to GS, that activation of adenylate cyclase that GS does produces cyclic AMP. That then is an incredible stimulant for smooth muscle cells to relax. We're going to use agonists at this receptor to see if stimulation of this actually causes relaxation. That agonist is going to be 5-CT, and you've seen that. We'll also use antagonists. This is SB, SmithKline and Beecham, SB 269970, and Lilly's LY215840 as antagonists to test if this is in fact mediating that relaxation. This is, for me, a really great graph because it gives all the messages I need to you, and that is that the 5-HT7 receptor really does mediate relaxation. On the x-axis, it's 5-CT, percentage of ET contraction, so all these tissues are contracted ahead of time. 5-CT in the vehicle, in the open circles, caused a beautiful concentration-dependent relaxation. LY272015, which I've not introduced to you, is a 5-HT2B receptor antagonist. And of all the 5-HT receptors that are around, this would be the other potential one that could mediate relaxation. It didn't do anything. By contrast, when I use either SB compound or the Lilly compound, I can abolish 5-CT-induced relaxation. What I'm not showing you and have published is that we use reams of serotonergic agonists and many other serotonergic antagonists to be able to focus pharmacologically and provide confidence that the 5-HT7 receptor is really that which mediates this relaxation.
So on that note, Stephanie, what is the specificity of 5-CT?
So it has affinity, high affinity, nanomolar affinity for the 5-HT7 receptor, but also for the 5-HT1A receptor. So those are the two that are together. What I know, though, is we can use 5-HT1A receptor antagonists and show way 100635 does not like him. There also aren't really a lot of 5-HT1A receptors in the vaccine. So. All right, so this is great. So we have in vitro evidence that says the 5-HT7 receptor is important. Right? But remember how I started my seminar, right? We found one thing in vitro, that 5-HT is a vasoconstrictor. And I hypothesized that then it should increase blood pressure in the body. Right? So in vitro to in vivo wasn't exactly smooth. Right? So what we have to do now is prove that our in vitro observation of 5-HT causing venodilation really occurs in vivo. So almost 3 years ago, we learned and helped purchase this Vivo 2100 that's part of the in vivo system. And Teresa, I know I saw you here. Where are you? She was here. She was really important to these experiments. We stood and talked in this hallway right outside this room about 3 years ago, and I asked both Teresa and Bridget, do you think we could use this machine to measure venous diameter or any kind of splanchnic diameter? And they looked at one another and said, well, I don't know why not. And they went to work. And to these two women, I will never be able to say thank you because they really went to work and figured this out. This is what this looks like. So we are going to take an isoflurane anesthetized animal on a heated platform, and that platform can move. This is the transducer that's going to allow us to visualize these veins, and this is how it looks. That tan square you see on this slide, that's the plane that the transducer is going to be able to see. So we're going to move that right around so that we can get to the superior mesenteric vein, the portal vein, the abdominal inferior vena cava, and its paired aorta. All right. One of the cool things about this is that an animal in this experiment can serve as its own control. What Bridget and Teresa discovered is that venous diameters don't change during the course of a typical experiment with no intervention. They also discovered that as a rat ages 8 to 10 weeks, at least during the time that we might think about giving 5-HT, the venous diameter doesn't change. So you have static measures you can take. You also have landmarks in the rat. So if you can know how to find the splenic artery and the splenic vein, then you can figure out where you are in this. It's a bit like reading the moon, right? I've had to get used to looking at these kinds of ultrasonography pictures. What that means, long story short, is that these animals serve as their own control. So I'm going to show you some data where you're going to look at the diameters under baseline, and then you're going to look at the diameters 24 hours after 5-HT has been given. Right? So let's see if I can make this work. All right. So many of you have seen this before. Focus here then on the portal vein. Right? It's tiny. You really can't see it. The abdominal aorta, which is here, the abdominal inferior vena cava. So these two are running parallel to one another. This is a still picture from the movie of now baseline. So you're going to see a change now when we give 5-HT. So baseline, watch the portal vein. So yes, Bridget, I know, I'm not trying to embarrass you. She came up and she said, it's a stovepipe. This animal should explode, right? It's just phenomenal. So let's see if I can make this happen again. And I don't know that I can. So it was just phenomenal in terms of the increase that happened. And certainly the magnitude to which it happened was not something we were prepared to see, right? So let's go ahead and quantitate those data. First, I'm going to show you what happens to blood pressure in these animals. She was able to put in I forget, did you do this with telemetry or with, this was telemetry, all right. So these data now are going to be as a percent of change from baseline, because remember the animal can serve as their own baseline. So it's mean arterial pressure, heart rate, systolic, and diastolic. The first half of each of these panels is what happens in the first 24 hours. That's at the point we're making these diameter measures. The second half of the panel, which would be here, that happens a couple days after we remove the pump. So if you look at the black compared to the white, the black is with 5-HT. 5-HT decreased blood pressure, increased heart rate as we expect, and of course decreased systolic and diastolic. When that's compared to control or receiving vehicle, when you took that pump out, then you reverted these, and in fact you had something of an overshoot in most of these effects. So in these animals, in which we're going to measure the diameter, blood pressure has fallen when they receive 5-HT. These then are the diameters. The SMV is the superior mesenteric vein, portal is portal vein, the abdominal inferior vena cava, and the abdominal aorta live right next to one another. They are only separated by a fascia of tissue between the two. What is very clear is there was venous dilation in the superior mesenteric vein and the portal vein, and over a 40% increase in the abdominal inferior vena cava. This animal, everything is exposed to 5-HT, but the aorta isn't changing. It is not dilating. Very consistent with what we've seen in the past. When you take now the pumps out, you revert these dilations. So right now, right, it's tempting to say that 5-HT is causing this venodilation, and that venodilation assists the fall in blood pressure. But what we need to show is that this is in fact 5-HT7 receptor dependent. So Bridget did these experiments. We're going to have the same output, right? These are going to be the diameters on the top, the cardiovascular parameters on the bottom, but now there's an extra group of animals. You have animals receiving 5-HT in the black. In the open bars are animals that received that 5-HT7 receptor antagonist, SB269970, before 5-HT was put on board. But it also did a really important control. This is saline, where both the agonist and the antagonist volumes were given, because we're now dealing with a venous-dependent event, right? And veins respond to volume. What we observed is that not such a good dilation of the SMV, right, but a really good dilation in the portal vein, and an even better one in the abdominal inferior vena cava. Compare that black bar to the white bar. That 5-HT7 receptor antagonist abolished venous diameter increase. Venodilation is gone. In these same animals, if you go to the bottom panel, what you see on the far left is that 5-HT reduced blood pressure, but that fall of blood pressure didn't occur when SB269970 was on board. So right now, both of these events, the fall in blood pressure and the venodilation, are mediated by the 5-HT7 receptor. And you want to jump to cause, right? You want to say this venodilation has got to be causing the fall in blood pressure. That's part of what we really still have to work out.
So, right? Stephanie, were you surprised not to see an increase in mean arterial pressure once you block the 5-HT7 receptor?
A little bit, and we've seen in some other experiments, we have seen an increase, because you're getting rid of this receptor that blocks it, and now, as long as your 5-HT concentration is sufficient to interact with the 5-HT2A receptors, then yes, we should see that. All right, so I said then, we sent this grant to NIH, and they came back and said, you use too much pharmacology. I'm a pharmacologist, what am I supposed to do? So after a lot of cursing and grumbling, we came back, and thank God for Elena Demariva, What she did is he helped us make this animal. This is the 5-HT7 receptor knockout rat. She took this gene and created 2 different knockouts for us. I want you to focus on the bottom one. This knockout has 2 different indels, and in this case, they are deletions of 11 nucleotides in exon 1 and 4 nucleotides in exon 2. And what that does is create a whole lot of stop codons, and this gene becomes non-functional. To the right, all I do is show you that we really did do the work, right, to use the founders. So there's a whole lot I won't show you here, and that boy, these animals just are a whole lot different from one another in baseline. Clinical chemistries are largely the same. This figure shows you that the body and tissue weights, they're really not different between the wild type and the black and the knockout. Here I've started to differentiate between males and females. So on the left-hand side in the green, you'll see the males. On the right-hand side, in what looks like gray but is supposed to be purple, are the females. Notably, females are, of course, smaller than male rats are, but relative to looking to retroperitoneal fat, perigonadal fat, kidney, liver, spleen, heart, and brain, they're just not different between one another at baseline. These are adult rats. So there you say, Stephanie, do the experiment. Give 5-HT to these animals. And that's just what Bridget did. So now these experiments, in the top are male and the bottom are female. It's exactly the protocol you've seen. On the y-axis is conscious mean arterial blood pressure. We have 5 days of control, 7 days of giving 5-HT, and then another 5 days of control. This is the same dose of 5-HT that we used previously, the 25 microgram per kilogram per minute in the Alzap pump. What is very clear is this wild type animal, which is based on a Sprague-Dawley line. It falls in blood pressure, be it a male or a female. If, though, you knock out the 5-HT7 receptor, now you do not have a fall in blood pressure. This is absolutely consistent with what SB 269970 did, and that we could not observe 5-HT cause a fall. Yes?
Is this with normal diet?
I'm sorry?
Normal diet? Diet is normal.
Yes, yes.
Okay.
Oh, absolutely. Yes, I'm sorry. Absolutely normal diet, the same one that our Sprague-Dawleys have normally been on. We have not yet challenged these animals with any kind of diet. So this
Stephanie,
is,
quick question also.
Yes.
Around 5-HT7 expression in tissue, what, first off,
I was going to do that later.
okay, but the other part of it is, how much variability do you see between animals? Or is it fairly consistent?
Okay. So, Bridget, do you want to, between animals here? So what I think I can do is tell you that when we've had different sets of knockouts and wild types, you look at this, right? We were really intrigued by this. The fact that losing the 5-HT7 receptor appeared to cause what was not a statistically elevated, that looked like an elevated blood pressure here, right? In different groups of knockouts, we haven't seen that, right? So there is at least some variability from baseline and blood pressure. Every knockout animal we've looked at, though, does not respond to 5-HT. So there's been no variability in that regard. Is there a particular variability you're concerned about?
Just, I'm trying to think further down the line in a human population, how much variability of 5-HT7 there is in tissue and what you might expect for a response.
Okay. So if you had to name the top places that the 5-HT7 receptor is exposed, the first is the brain. The next would be the intestines, immune cells, smooth muscle. Those are the places that most people would believe that 5-HT7 receptors are important and are functional. There's very little pharmacology around 5-HT that is therapeutically designed. Right? Of anything that's happening in the literature right now, anxiety, schizophrenia, and depression, as well as dysfunctions of the circadian rhythm, have been the target for 5-HT7 receptor antagonists.
Right.
Okay? We're after a different thing. Yes, Karen.
What age are these rats?
So these are just about 12 weeks of age. We have gone up in Estes, Karen, to animals as old as 9 months and been able to demonstrate that 5-HT still causes a fall in blood pressure. But we haven't gotten these girls. We use them before they get that old, right?
Did you see a difference in baseline between the ages?
No, and we need to do that experiment. We need to just let them grow and understand what would happen, because you're right. This is but one snapshot in time that we're taking your baseline. Very important. All right, so far so good. All right. What was so interesting to me is what was happening in the vasculature. This is really where this project was born. Here I show you a tracing that comes from a chart, a file of an experiment that I did, where on the force, this is what the vessel did. That's the y-axis. On the x-axis, while it's time, I show you what different interventions that I use. So this is an abdominal inferior vena cava, right? One that went 40% bigger in the wild type or in the 5-HT versus not. We contract with endothelin. And when you give 5-CT, which, as I told Rick, has affinity for the 5-HT7 receptor and some for the 1A, you see a beautiful relaxation. When you add this antagonist, which we know can abolish 5-HT-induced hypotension, I've never seen this before in my life, and I've done a lot of tissue bath experiments. The antagonist reversed it right there. That's unusual for me to be able to see. This 5-HT7 receptor antagonist abolished the 5-CT-induced relaxation. What we did at the end here is to add forskolin. Forskolin, you know, is an adenylate cyclase activator. And one of the things we needed to be able to show is that our knockout vessels haven't lost the ability to respond to a cyclic AMP stimulus, and that's what forskolin would do. So this is what happens in wild type. So now we're going to take a littermate, and this is what happens in the knockout. Again, we contract with endothelin. When we add 5-CT, it didn't relax, and in fact, what you have under here is a contraction. These oscillations, you simply cannot guarantee when they're going to happen. And of course, they happen at the most unfortunate time. So this is where 5-CT SB, when we added it here, I could see no quantitative difference. And we did have cleaner experiments. The most important part of this here is that when you added forskolin, you saw a relaxation. So veins from knockouts can still respond to a stimulus of cyclic AMP generation. Okay, so that's not a reason that these animals aren't falling in blood pressure to 5-HT. So there's even more interesting work you can do. Now, that's actually just quantifying this work. We'll go to here. So that's what veins did. Here, we compare what the aortas do. On the left is the male, on the right is the female. Knockout is always the solid symbol. The aorta, or arteries at this point, are not tissues, I believe, are 5-HT7 receptor responsive. What you see here is that 5-HT-induced contraction is simply not different, whether you're a male or a female, between the wild type and the knockout. That's consistent with what we've seen before. Now we're going to take the vein that lives right next door to this aorta. On the bottom, this is the abdominal vena cava on the left from the male, on the right from the female. Look at what happens in the wild type. You have a small contraction to 5-HT in the wild type. When you remove the 5-HT7 receptor and the knockout, now you amplify that contraction substantially. So you're moving from here to here. That suggests that in the venous circulation, but not in the arterial circulation, the 5-HT7 receptor is a brake to contraction, right? Because of its constant production of cyclic AMP. So does this make sense? Right? So we're revealing what 5-HT would normally do by taking away a 7 receptor. Right? Vessels have many different receptors of many different kinds, and what you get out of it, in this case contraction, has to be a synthesis of those different things. That right now is our hypothesis, and I can tell you we can shift this curve with the 5-HT2A receptor antagonist, right? It has a PKB value that's consistent with the interaction of 5-HT with the 5-HT2A receptor at that point. So these two circulations are pretty different from one another. And that's interesting.
Why does the vena cava dilate in vivo if it contracts in vitro?
Well, but it did. We haven't done that in the knockout.
Well, but here, just looking at the control, your wild type vena cava contracts the serotonin.
At high concentration.
Right, but
And what we think we're achieving when we give it in vivo, we're here.
okay, so these aren't pre-contracted.
No, these are
Right.
from baseline. I apologize.
Yep,
Yeah.
yep.
Okay.
Yeah, that's the
But
difference
now,
between
what
your
you're
previous
getting at
experiment.
is, this is one of the coolest experiments I think that Bridget did. What she did is to try to get into this idea of, could we see the same kind of pressure response in vivo? So measuring these conscious blood pressures, this is minutes, and gave increasing doses of 5-HT from 12 to 25. This is where we're standardly. 5, 50, and 100. This is what's happening in the wild type. So we see that fall here. We do see an increase in blood pressure here. Right? That's not something that we'd seen before. Look at what the shifts in these curves are. Right? When you remove the 5-HT7 receptor, as you do in these open circles, now what you've done is taken away the brake on blood pressure. And right now, the primary place we think that's happening is in the venous circulation. Right? So let me summarize, and then we'll get to the drug discovery part of this. And that's really where I need Edmund and Tom to come up. So 5-HT reduces blood pressure. Doesn't increase it unless you're a knockout and at a certain dose. It relaxed isolated veins, and it did so because it could activate the 5-HT7 receptor. That receptor, when activated in vivo, served not only venodilation, but a fall in blood pressure as well. Everything that I've shown you, and we have done some work in the knockout with venodilation, suggests that the 5-HT7 receptor is essential for this. Right? So what? Well, we have causes of groups of people that need treatment. Right? In the world of hypertension, essential hypertension was just redefined a year ago. So if you read or watched CNN, you'll know that 130 millimeters of mercury systolic and 80 millimeters of diastolic is now hypertensive. That makes 46% of our adult population hypertensive. Right, that's scary. I bet you there are a lot of phone calls to doctors, right, the day after that occurred. So a lot of people have essential hypertension. There's also this form of hypertension called resistant hypertension. And these are individuals who can be on 3 or 4 medications. Right, so they should be on the floor, but their blood pressure is not following. Depending on who you talk to, estimates are anywhere from about 8 to 14%. Of the hypertensive population would be those individuals who, no matter what we do, their blood pressure doesn't fall. Last, and what is really going to be the focus of where I think this goes, is heart failure. So this is simply having a failing heart. Hypertension is something that drives people into heart failure. If you talk with clinicians, they will tell you the 2 greatest problems they have are treating atrial fibrillation and heart failure. This is the #1 reason that people are rehospitalized because of heart failure. In the clinic, most clinicians would say one of the only ways that I can help with heart failure is to reduce the work of the heart, and I can do that by reducing blood pressure. So there are very real reasons for wanting to develop something like a 5-HT7 receptor agonist in the face of the idea that cardiovascular disease, which includes D3, is still the cause of more deaths than cancer and chronic low respiratory disease and accidents combined. We have a challenge to be able to meet.
Stephanie,
Yes.
is hypotension ever a problem
Oh, yeah.
too? Because I know in drug discovery, you can design something to be an antagonist or an agonist, and can be the opposite. So
It absolutely. There is a clinic in
Elderly
Vanderbilt dedicated to autonomic failure and hypotension. I have been trying to talk with people about getting measures of 5-HT to understand if there are
Yeah.
dysfunctions in 5-HT handling or metabolism in patients that have hypotension.
so would this be a viable, you don't know if this would be a viable target for that either, or?
Well, because there, I think what we'd want to do is have a 5-HT7 receptor antagonist
Right,
to be able to
right.
block that. No.
So when you're designing drugs, sometimes you think you're making an agonist.
Yep, and you actually don't have it. I agree. So I would want to talk with David Robertson and Italo Biagioni, who are head of the orthostatic clinic, to understand if that would be. One of the places we find ourselves is not a lot of people talk about 5-HT in the ways that we do. So
Yeah.
there aren't a lot of clinics and there aren't a lot of basic research doing this kind of thing. So that leaves us a lot of space to go, Jamie, but it also leaves us with not a lot of people to play with.
Yeah.
So it's a great idea. All right, so this is Edmund's slide. So his goal as the MDG that he and Tom and I have is to develop a 5-HT7 receptor agonist. And where we are right now is analog design and synthesis. The hope is to be able to take the compounds that Edmund and Tom make and use in a cell-based assay. And I know Tom has done some of these cell-based assays. Do you want to talk about what's going on, or do you just want to let it go? So we're working on replication, and that's where we are, where we might have some compounds that we can move forward. I've got a great bioassay that if Edmund tells me he's got a compound and Tom tells me he's got something that would stimulate cyclic AMP in his assays, I'm going to test it in a vein. Right? And I'm going to test it in an artery, and I know if it's an agonist, it will relax the vein, and I should be able to block that with SB26997L. So those are the cell-based assays that we take through before we go through something like in vitro ADME. The goal then would be to do rat PK and to ultimately look at the efficacy. We have a great bioassay for that as well. We have blood pressure as an endpoint. And we have a knockout rat that we can be able to test these in. I promise I'm almost done. And then this slide is for you. This comes from the Human Protein Atlas as to where the 5-HT7 receptor is. I'll read these to you because it's so small. Brain protein is on the right. Endocrine tissues is the purple. Bone marrow and the immune system. One of the most interesting findings is the idea that 5-HT7 receptors are on virtually every immune cell. 5-HT7 receptor activation appears to be anti-inflammatory. That's a bonus, right, for any kind of drug we could potentially make. The other place we have to worry about is the gastrointestinal tract, the kidney, urinary bladder. We have to apparently worry about males more than we do women. What's stunning is the vasculature is nowhere here, but that's where it is. So here are some of the opportunities that Edmund has used for the scaffold hopping. There are programs, specifically in Spain and Italy, that are dedicated to making 5-HT7 receptor antagonists for the treatment of some of the CNS disorders I described earlier. So RA17, AS19 as an agonist, some of the Esteve compounds, these are what he's working on to ultimately being able to make some of these compounds that then we can take through. Okay. So I want to thank you for listening to me. I would love to hear what you think about this. I want to argue with you about science and think about where we could possibly go. So thanks so much for listening.
I have a naive question and naive idea. So we all know that serotonin is like released from the EC cells in the gut and it keeps releasing like crazy. So there must be somehow the mechanism that transport the serotonin from the gut movement into the circulation, because from your data, serotonin is not come from the nerves, right?
No, it doesn't appear to be. So that would be really through the platelet. So platelets contain millimolar concentrations of serotonin. They do that because they have the serotonin transporter on the outside of it. So when 5-HT is released from their enterochromatin cells, think of platelet as the Hoover vacuums for 5-HT. They have an incredibly efficacious transporter that lets the platelet bring serotonin in. Wherever, though, the serotonin transporter is, 5-HT can get in. Right? So the platelets are the only place that 5-HT can be. We've done the experiment where we've infused 5-HT into the rat and found that multiple tissues, I'm not finding the word, they gather 5-HT. And when you knock out the serotonin transporter, they stop doing so. And what I can't tell you is that I've gotten rid of every single platelet that might be in that tissue when we go to measure 5-HT, but it appears that the sequestering of 5-HT is not the function of the platelet alone. That, though, is where I think it primarily is. This is not to say that 5-HT is nothing outside the platelet. There's always free 5-HT, right? That's measurable. One of the problems we have in the field is really measuring that with confidence. There are papers dedicated to saying we don't believe any report that says you can measure free 5-HT, because the process you have to go through to do that might make platelets fracture, and then you've swamped what could be free 5-HT. So, does that answer your question? Right? That's where it comes, and that's how it gets there. Okay? Yes, Rick.
So there are, what, 27 different serotonin receptors?
At least.
And
Well, 14.
And you're making an agonist, which has the potential to turn on all 14 of those receptors. So what are you going to do to keep that from happening, and which ones do you have to worry about?
Okay. So probably in terms of drug discovery, the one we have to worry about most is the 5-HT2B receptor. And because 5-HT relative to the 5-HT7 receptor has the greatest affinity for the 5-HT2B receptor. And this has been the receptor which has stopped drugs ever being developed. So if you've heard of fen-phen, do you remember this duo taken for medical management of obesity? Primarily obese women started turning up with pulmonary hypertension and dying of valvular failure. It was because of activation of the 5-HT2B receptor. So Tom has got a 5-HT2B receptor cell line that these will be tested along with. Pharmacologically, I think we have to worry about overlap with the 5-HT1A receptor the most. One of the goals, though, is to keep this drug not entering the CNS. right? So I want it to stay here. I do not want it to go this way. I know that's reversed for many people, and Edmund tells me that it's easy to do, right?
Easier than the other way.
Okay, so that's really would be the goal, is to try to see if we can do this. And we're just gonna have to test.
Yeah.
So we get something that can interact with those amino acids in the 5-HT7 receptor that make it specific for it.
Yes, you're basically looking for a non-brain penetrant 5-HT7 agonist that doesn't hit 5-HT1A or 2B.
Yes.
So let me ask.
Do you have one?
Let me
I
ask
think
one
you
further
do. I
question.
think you
We
do.
do have one, right? But, so let me ask one further question. If you don't hit 5-HT1A and you don't hit 5-HT2B, is CNS penetration acceptable for heart failure?
Oh, Edmund, I'd have to think about that. In terms of the.
The absolute desire is the profile you described. But if you have half that profile and heart failure is your indication, what's acceptable?
Potentially, but I'd have to go back and really think about how 5-HT, there are sites of the 5-HT7 receptor in the brainstem that control cardiac rhythm, to understand what activation of those receptors could possibly do. That's an interesting idea. Yes. Peter.
So I've always wanted to ask you this question, and it seemed appropriate. So 40 plus years ago, when there was no idea that there were multiple types of serotonin receptor, there was an article published showing that propranolol was a really good competitor for serotonin and brain synaptons. Does propranolol affect your serotonin relaxation? Because that would be relevant to people being treated for all of these conditions.
So Peter, we've never exactly tested that idea. I will say two things. First of all, propranolol, right, is related to some of the olols, like bucindolol, which have affinity for the 5-HT1A and 5-HT1B receptors. So that's really part of how that field started, is with the knowledge that it could do that. One of the considerations Patrick had in his thesis is, could we cause a fall in TPR because somewhere epinephrine was being released, like from the adrenal, and that is a beta receptor antagonist dependent event. We never got to do the whole animal experiments in that way. So I have to say I don't have enough information for you. One of the cool things though is if you go back to the original finding from Irvin Page, who helped discover 5-HT, he was the first to show that 5-HT causes hypotension in the human. And so what we have is something that's possible. What's so fascinating to me is this story has so many different chapters, right, where it's gone through. I'm hoping we're at an interesting one where we can actually find this agonist and really test it. This, everything I've said, could not have been done without Greg, without Bridget, without Edmund, without Tom. It has certainly been a labor of love for me and something I've really enjoyed doing. So, yes.
So tell us about heart failure. We haven't heard really much about heart failure, and that may be your best clinical indication.
So what would you like to know about
So
her?
why
Recognizing
do
this is not,
you really think 5-HT7 receptor agonist will help in heart failure?
because of reducing what would be both preload and afterload, right, on the heart. Right,
So you're lowering blood pressure for one thing, but you can do that with all the current antihypertensives.
you can.
So the idea would be that by causing venous dilation and the splanchnic circulation, you're going to reduce the inflow.
But it's part of it. So one of the things we haven't seen, right, with the 5-HT7 receptor agonist is a fall in cardiac output, right? So I'm not sure we're going to get that piece of the puzzle. Greg and Bridget, jump in wherever you want to. Greg has probably thought more about heart failure than I have.
I thought I saw an increase in heart rate.
Within the first 24 hours, but then it comes back.
Okay.
What we don't know, there's no role for the 5-HT7 receptor at this point yet in directly changing heart rate or inotropiscity of the heart. Right? That's
No, I
not been
think the heart
discovered
rate
in the rat or
was
the
going
In
up.
an isolated heart or in?
And it was not a lot has been done in an isolated heart. In the rat, the few things that I could find suggest that it's out there and that there's not a lot of the 7 receptor outside of the vasculature that supplies the heart with blood. So,
How about the intestinal function, gut function?
so we have, I bet your lab took parts of these intestines. We still haven't heard what happened. I'd really like to know, right, what happened from these. No, I would offer this model to anybody who wants to be able to take these and test different ideas about this. We have done no experiments relative to gastrointestinal function.
Stephanie,
Yes.
I did, like, on your graph with the knockout wild type mice, I did see that the females that in recovery have higher.
Yeah, they do. They do. And one of the things, let's go back to that, Anna,
Yeah.
that we have never been able to, and Peter, I know you've asked me this question too, right? They do come back.
Yeah.
Right? And different groups of animals come back differently. Physiologically, right, if you think about homeostasis, an animal does not want to have this lower blood pressure because it's reducing flow to tissues. So some animals may recover better than others. What we know is that serotonin is still there. Serotonin is still present, it's still functioning in the pump. We can still measure an increase and the veins still relax.
Wouldn't this be like concern about when you're doing pharmacological intervention and people are known to that?
Well, so one of the things I can tell you, we took, understood, is we took DOCA animals. Those are the first we did. We gave 5-HT for 30 days. Right? And we're able to show a 16 millimeter reduction in mean arterial pressure through those 30 days. So though it recovered some, right, in those particular, it did not recover absolutely. I do not yet have a good handle on why groups of animals are different from one another in terms of their recovery. It is an absolutely relevant point.
So it looks like, yeah, so basically it looks like tachyphylaxis.
It is, but I can tell you from the venous level, it's not.
And we see that often with SD rats. When I take off the 5-HT stimulus, I will see an increase in blood pressure.
Well,
It almost is a ramp
I'm
up.
talking about the recovery during the stimulus,
Yes.
not the
Yes.
overshoot.
Right. Sure.
Yes.
So they're both probably related.
Right.
So does the receptor downregulate?
So why we thought about that experiment, I took tissues from animals that were right here and could demonstrate in vitro that they relax perfectly normal to 5-HT and 5-CT. That experiment comes with the problem though, right, that I've washed the tissue for some time before I get to it.
Yeah.
So if it desensitizes, then it resensitizes really fast.
When you say sometime, you watched it for half
Minutes.
hour?
Yeah.
Oh, no, that's,
Yeah.
then I wouldn't worry too much about that.
With your,
I worried about it.
with your super duper mini pump, which you can alter the dose rate, what happens if you give 12 hours of infusion of 5-HT, 12 hours off, 12 hours on, 12 hours off? Can you maintain the, I mean, I don't think you've done it, but would be an interesting experiment to see whether you actually get maintained.
So I've done something like that. So I have a conscious animal with a radiotelemeter in it, and I put a chronic vein line in it, and I give it 5-HT, and I stimulate it every other day with SB, which is the 5-HT7 receptor antagonist. So blood pressure is down. I hit it with the antagonist at different time points of that challenge of 5-HT infusion, and I can show recovery of blood pressure back to baseline.
So it still happens.
So it's still, so you still see a fall in blood pressure with 5-HT, and at say day 2, and then day 6 of infusion, I infused it for like a week of time, and I can show that I can block that 5-HT7 receptor fall in blood pressure at those different time points, showing that the activation of the receptor is still there during during the time of infusion.
Yeah, but what
Your
I'm saying
experiment is different.
it's slightly different, right? What I'm saying is that this tachyphylaxis,
Right.
as Rick referred to it, which, I have no objection to the word, Rick, could be prevented if the tissue wasn't exposed to the agonist.
So
Which is why I
does
say 12 hours on, 12 hours
Does
off.
intermittent exposure work
So-
better, for example, than continuous
It
exposure?
was turn it on, turn it off,
Yeah.
turn it on, turn it
Yeah.
off. I don't know.
Or you take one dose in the morning and the level is high in the morning,
Yes.
and the level drops overnight, the receptors resensitize and you can get a good, who knows?
Yeah.
Right.
So interesting things, and then I know we're out of time about the 5-HT7 receptors that really make me want to do this. It does seem to be prone to desensitization, say the way many others, and it's been described as being a pre-coupled receptor. So I'm really interested in knowing what kind of constitutively active receptors there may be. So come talk to us anytime. I'm delighted to share our science with you, and thank you for your time.